topic for today, which is the complement system. And this is a system of plasma proteins that mark pathogens for destructions. Mark pathogens or code pathogens for destructions. I had told you in earlier lectures that there are two ways of marking, pathogens or marking cells that are infected with some kind of pathogen. One is to use an antibody for that purpose, and the other is to use a complement. So the complement system is important because this is the first to come into play, and the antibodies production is to happen later because the adaptive immune responses are generated at the end of infections when the innate immune system is not capable to do that. So the adaptive immune system, the complement system, these are plasma proteins. About 30 different plasma proteins are synthesized in liver, and these proteins are the major weapons of the innate immune system, or you can say the primary weapons which are fired is they see some kind of infections there in they are present in the blood, lymph, and extracellular fluids. So it means their distribution is, very much in all the body fluids, okay? In the lymph, in the blood, in all extracellular fluids, you can find these complement proteins, okay? And these are actually serine proteases present in inactive form, and we call them zymogens. What are serine proteases? These are enzymes, proteolytic enzymes, that are going to act upon all the peptides to, detect peptides that have serine, this particular amino acids at various, in various pockets, and cut all those peptides right from there, okay? So we call them serine proteases or some other amino acids, leucine or some other, you can say proteases are named like that. This is the general name. So these enzymes are not active when they are made by synthesized inside the cells and secreted out into the blood. So it means that they are normally synthesized inside different cells of the liver, and then these these enzymes in an inactive form are supplied to the blood, or they are transported into the blood, and they are one of the first weapons to fire by the innate immune system. They are activated when they see some kind of pathogen who does not belong to the body. So these are the first, this is the first, weaponry to be used by the innate immune system against all those aliens which enter the body. Infection triggers activation followed by a cascade of enzymatic reactions. The infection activates these proenzymes or zymogens. Okay, the inactive form is turned into an active complement once there is infection, once there is entry of the pathogen. And what do? Actually, they coat bacteria and extracellular viruses. And how do they coat it? We call this process as complement fixation, okay, in which the complements would attach to pathogens. For instance, the surface of bacteria or to a virus, for instance, the complement can attach itself to these different pathogens. This is complement fixation, or coating of the complement of the pathogens with the complements is called complement fixation. This is, for instance, complement fixation here. We would talk about all the complements, but C3 or the complement number 3 is the most important complement that you should remember. Because some people may have deficiency of this complement 3 or C3. When they have deficiency, they are prone to develop different kind of infections. Very prone to develop bacterial infections, for instance, because on bacterial surfaces, C3 is the complement which would be attached to the bacterial surfaces, or it is used for fixation. Okay, so for instance, this is C3, the complement, and when this complement is cleaved into two parts, one is called C3b and the other is C3a. This complement is activated. Now, this activated complement, one part of the activated component, which is the bigger part, which is C3b, it attaches to the bacterium, When it attaches to the bacterium, this is called tagging or coating or fixation of the complement. While the other complement, other part of the C3A, it is, a chemoattractant, you can say, C3A or C5A, both of them are chemoattractant. And they recruit phagocytes to the site of infection. Because there is infection, this is a bacterium which has caused infection. And the bacterium now, has been coated with a part of the complement, the bigger part of the complement, which is part B of which complement? C3b. So the bacteria, this would increase the phagocytic process of this bacterial species, because now the body has to get rid of it. So there are mechanisms to be used by the body. One mechanism is to recruit these phagocytes to come here at the site of infection and just, engulf on the tagged bacteria and get rid of them. Okay? Another is to use these different types of complements in order to make a structure and penetrate the membrane of this bacteria and destroy it, or just, digging hole into the bacteria. That is another mechanism which also can be accomplished by these complements when they come together, So on one hand, they are going to facilitate with the process of phagocytosis, and on the other hand, they are also directly killing, the microbe, So they take bacteria for destruction by phagocytes or by making a protein complex, which is known as the membrane attack complex, or MAC. Okay, and the other part of the C3A is, going to recruit phagocytes. So now you can understand that why C3b is very much, or C3 is very much important. Okay, because if there is no C3, or people who genetically do not have C3, because this is being encoded by a gene. If there is some mutation that is not, produced inside your body, then this process of phagocytosis would be very less efficient. And on the other hand, in the absence of this membrane attack complex, which cannot be found without C3, these would not be formed, so people would be very much prone to develop bacterial infections. And that would be very fatal in many cases. So what's the secret of C3 being so important? Okay, this is C3 here, and C3 is cleaved into two parts, as I told you, which is C3b and C3a. The smaller part is called A, and the larger part is called B. Okay, so what is hidden there with C3? Actually, if you look here, here is a bond here. Here is sulfur, carbon, and oxygen. This is a thioester linkage. Okay, so the thioester linkage, when C3 is in inactive form, synthesized in the liver, this C3, this thioester linkage is hidden inside, this particular structure C3. But when you cut it into pieces at certain places, then this thioester, bond, this is exposed. The C3, the smaller C3A smaller part is detached, exposing this particular linkage, which is the thioester bond. Okay. Now, what is the thioester linkage actually? You can see that this is the linkage of, carbon with oxygen having a double bond and a sulfur. Okay. You can say that these are some kind of organic sulfate compounds, some kind of that compounds that have this kind of linkage. So what is the significance if this thioester linkage now has been exposed, when C3 is cut into two pieces? The significance is that once this nucleophile is exposed as a result of this cutting, it becomes prone to be attacked by other molecules, and the best molecules for these purposes are, for instance, water and some kind of amino acids that can readily come here and make a bonding with these, thioester. Okay. For instance, you can see that it can be attacked by water, and when the water attacks this particular structure, there is a lot of room to adjust the water molecules right here. And you see that the water molecules are, adjusted in the form of hydroxyls. You can say OH and H on the other side. Okay. And on the other hand, this can also be attacked by amino acids. The NH2 group can also attach here to this. So if it is, the water molecules attach to the thioester linkage in C3, it would result in a soluble C3b. Okay. But if an amino group attaches itself with the thioester linkage, it usually happens on the surface of the pathogen, and in this doing so, this is some kind of a covalent linkage that or bond which is made between C3b and the surface of the pathogen, So in this way, when C3b, it binds itself to the surface of the pathogen, it is called coating of the pathogen. It marks the pathogen in this way, that it would just make a covalent linkage. And why would the covalent linkage be made? Because the thioester linkage, it is prone to make bonding with other molecules, particularly water and amino acids and amino group. The water also is abundant on the surface of the pathogen, and these molecules are also found on the surface of the pathogens. So there are more chances for C3b to become, bonded to the surface of the pathogen and coated there. And that is the process. That is the start of the process. That how it starts, how the complexes are made, or how the pathogens are killed. That's the first thing that should happen if the pathogen has to be killed or to be neutralized inside the body.